This video will be talking about the nephron, which are the functional unit of the excretory system. That is, they play a major role in filtration and bringing about or the formation of the urine. So we will be talking about the structure of the nephrons in detail in this video. Each kidney, when we are talking, it has approximately 1 million of nephrons, which are the functional units. And these kidneys were discovered by Malfigi. He was the scientist who discovered kidneys, and the structure of the kidney was given by Bowman. So these two scientists are responsible for allowing us or letting us an insight into the structure of the nephrons. When we talk of the structure of the nephrons, it is actually made up of two components, that is the glomerulus and the renal tubule. The glomerulus and the renal tubule, that is the Bowman's capsule or the renal tubule we call it, they together form the nephron. Glomerulus is a network of capillaries, and these capillaries are formed by the efferent arteriole. As you can see over here, the efferent arteriole enters in over here inside, and it is actually a branch of renal artery. Now the renal artery carries the oxygenated blood from the heart to the kidneys, and it is for the purpose of nutrition and cellular respiration. Now at the same time, the blood from the glomerulus is carried by the efferent arterioles. Now it has a lesser diameter than the afferent arteriole, which increases pressure, which is important for the formation of the urine. Glomerulus along with the Bowman's capsule is called as Malpighian body. So glomerulus along with the Bowman's capsule, it forms a body which is known as Malpighian body, or another term for it is renal corpuscle. There are two terms to it, Malpighian or renal corpuscle. Malpighian body or renal corpuscle. So these form the part of the nephron. Now the blood filters out through the capillary loops. As you can see over here, the efferent arteriole then forms a system, and over here the Bowman's capsule also. Now what is happening over here is actually, when we are talking of the structure, the blood filters out through the capillary loops, through the glomerular mass and podocytes into the urinary space. Now podocytes are specialized epithelial cells. The tubular system, that is the tubules, continue into a highly coiled network called as proximal convoluted tubule. So over here, this Bowman's capsule, the podocytes are present over here. The Bowman's capsule leads into the proximal convoluted tubule, which is a highly convoluted structure. Convoluted means it is having lots of bends and turns, like this it is having. So the proximal convoluted tubular structure, that is PCT in short we call it, and the terminal end of the PCT, that is the terminal end, is less convoluted, and the cells which are present in the PCT, that is they are simple cuboidal brush border epithelial cells. The cells are specialized for absorption and secretion. So over here in PCT, we find the cuboidal epithelial cells which are brush border type. Now this PCT actually helps in the filtration, and then we'll be studying about the tubular system in detail. We are studying the anatomy in general of the nephron. The PCT leads to a hairpin shaped Henle's loop. Now this Henle's loop has two limbs, the descending loop, that is the descending limb, and the ascending limb. Both constitute the Henle's loop. Now it enters into the medulla. Actually this Henle's loop, this region is in the cortex area, this region. Up to here it is in the cortex, and now it has entered into the medullary region. That's why we see medullary striations. And it has a descending and the ascending limb. The descending limb is almost in continuation with the PCT, and over here the cells have very less villi over here. Brush border cells are reduced. And the ascending limb then finally leads to distal convoluted tubule, which is known as DCT. Now it actually helps regulates the pH of the urine. The cells which are lining the DCT are simple cuboidal cells. DCT of several nephrons open up into a single tube, which is known as the collecting duct. So this enters into the collecting duct over here. The collecting ducts join and open into the renal pelvis through the medullary pyramids in calces. Now when we talk of the different parts of the nephron, the Malpighian corpuscle, that is the glomerulus, and the Bowman's capsule together, the PCT, that is the proximal convoluted tubule, and the DCT, that is the distal convoluted tubule of the nephron, is situated in the cortex region, whereas the loop of Henle and the collecting ducts are in medulla. So this region, as I told you, it is present in the cortex region, whereas this region is present in the medulla region. Now when we talk of the nephrons, there are basically two types of nephrons. In most of the nephrons, Henle's loop does not go deep inside the medulla. As I have told you over here, the Henle's loop enters into the medullary region. Now when we talk of the types of nephrons, these nephrons are of two types. The first is the cortical nephrons. Now, when we talk of the cortical nephrons, in these type of nephrons, the Henle's loop is very, very long, and it reaches down to the medullary region very deeply. And the others are juxtamedullary nephrons. The other is juxtamedullary nephrons. So as told, cortical nephrons, most of the nephrons, the Henle's loop is short over here. Henle's loop short. Second important point is they do not reach, or they reach only up to medulla. So just reach up to medulla. Those will be known as cortical nephrons. Now, other nephrons, other type of nephrons which are known as juxtamedullary, Henle's loop is long. Henle's loop will be very, very long, and it will reach deep into the medulla. These are actually involved in filtration. So the juxtamedullary nephrons are primarily involved into the process of filtration. Now, the efferent arteriole, which is coming out from the glomerulus, it forms a network around the renal tubule called as peritubular capillaries, and a small vessel of this network, that is, it is running parallel to the Henle's loop, forms a U-shaped vasa recta. Now, vasa recta is absent, or maybe vasa recta over here, that is the third difference over here. The vasa recta may be absent or highly reduced in cortical nephrons, whereas in the case of juxtamedullary nephrons, it is very prominent and well developed. Now, when we talk of the vasa recta, it actually helps in the reabsorption and secretion between the blood and the inner lumen or the cavity of the nephron. When we talk of the vasa recta function, they help in passing the blood into the renal vein. So from after the filtration, they help actually passing the blood into the renal vein over here. They also help in the maintenance of concentration pressure. So vasa recta is over here. This is vasa recta, and it helps in maintaining the pressure concentration or concentration gradient. We'll be studying about it in detail about the functioning. When we talk of the function of the nephrons, now actually when we are talking of the nephrons, it is in broader terms, or in more elaborate form, we talk of kidneys. So they help in maintaining the fluid balance of the body. Another important function is elimination of the nitrogenous waste and maintenance of acid-base balance of the body, removal of the mineral salts, drugs, bacteria, etc. They also help in maintaining arterial blood pressure by a hormone which is secreted, that is known as venin. Venin is a hormone which helps in regulating the arterial blood pressure. So the major structure, that is nephron, is having the glomerulus, the Bowman's capsule, the proximal convoluted tubule which is lined with the brush border cells, then the distal convoluted tubule, but the proximal convoluted tubule leads to the Henle's loop, which is followed by the distal convoluted tubule, which is made up of the cuboidal epithelial cells and is less convoluted. The distal convoluted tubule joins into the collecting tubules, and many collecting tubules join finally and move out through the renal pelvis to form the ureters. Henle's loop has the glomerulus consists of efferent arteriole and the efferent arteriole. The efferent arteriole has a lesser diameter as compared to efferent, because the blood will flow with great pressure, and this great pressure enables the proper filtration of the urine. As efferent arteriole reaches down through the Henle's loop, it forms another important structure, a system or network of fine capillary that is known as peritubular capillaries. And out of these peritubular capillaries, vasa recta is an important tubular system which actually helps in maintaining the secretion rate of secretion and the reabsorption within the capillary system. Then there are two types of nephrons we have studied, that is the cortical nephrons and the juxtamedullary nephrons. The cortical nephrons have a short Henle's loop. They reach up to medulla, or sometimes this Henle's loop may be reduced, Then also they may be having vasa recta, which may be absent or highly reduced. Juxtamedullary nephrons, in which the Henle's loop is quite long and they reach deep down inside the medullary region of the kidney. Also in them the vasa recta is very well developed, because these are actually taking part in the formation of the urine.